Hi, everybody. Welcome to another episode of the Labiotech Hangout. So this month we have the founder and CEO of PILI here. Hi, Jeremie.
Hi, Philippe.
Glad to have you here. So PILI is a synbio startup based in Paris and Toulouse. You're developing the next generation of dyes and pigments. So especially for the fashion industry, it's like the second most polluting industry worldwide. And how you're doing it? You're producing it with a bio/industrial biotech approach, so microbial fermentation instead of chemical synthesis. And so you have founded a few years ago, have a team of now more than 10 people, and you just closed a nice round of 2.5 million euros. So can you start by just telling me a bit more about this round, who you raised it from, how long it took you?
Yeah, sure. So it was not easy to close this round because we don't have so much experience, and we are at the crossroad of several sectors. Biotechnology, which is kind of technical and complex, but also specialty chemistry, and finally textile. So it's not easy to combine all these sectors. So basically, our investors are different kind of investors. So we are happy to gather both venture capitalist funds, but also business angels. Some are experts in particular in the textile industry. But also we raise a part of this round thanks to crowdfunding, which is also very important because it shows that people are very interested about this kind of innovation, especially for the fashion industry. So we feel support both from industry and consumer.
That's great. What was the split between crowdfunding versus venture?
Crowdfunding is approximately a third of the round.
Okay, that's great. And if you take a step back, so you said it's difficult because you're in different niches, but how is more the financial environment or the risk taking for clean tech? I mean, it seems like there was a big hype in the 2000s. Some VCs lost some money. So yeah. How is it right now?
Well, I don't know exactly because biotech it's not an homogeneous sector because you have biotech for Earth, etc.
Yeah, but I mean, I'm talking about the non-health biotech, like clean tech.
Okay, yeah, it's not easy because you have very few specialized investors, and product development time is generally very long. So the problem is for this kind of technology that classical investors are looking for return in the 5 to 8 years. So it's really difficult at the beginning of this kind of industrial biotech project to engage ourselves on a specific timeline. That's why this kind of project needs other type of investors. So it can be private investors, business angels, it can be corporate investors. At the beginning, for seed stage, for instance, we really need actors that trust us and that we don't need a fast return.
Yeah. Yes, that's what you did, and yeah, sounds challenging. So first, congrats on closing that round. I guess will help you for the next 18, 24 months, I guess. But let's take a step back, really at the very beginning of PILI. I think you have a really interesting story. You're one of the few startups in Europe or even worldwide to be born from a biohacking space, or La Paillasse in Paris. So can you walk me through it? How it started? How was it? Go back to the history.
Sure. Well, it started with curiosity. and patient, because one day I went in a cave in this biohacking space La Payas. At this time, it was not based in Paris. It was based in a suburb of Paris, and it was in an old building. They work in a cave with hackers, and I think I was at this time the first guy coming with a with a with this kind of suit and tie, because I made an internship in investment banking at this time. So
yeah, because you don't have a biotech background actually.
No. I made a business school in Toulouse. Yeah. So I was just curious about biotech, and on the other hand, my co-founders that are biologists made a workshop for children and for curious people that want to know how to make ink from sugar thanks to microbes. This is what we do. We produce renewable dyes from sugar thanks to cellular factories that are microorganisms. And at this time, yes, it was just a workshop, but a lot of success. The public was in love with the concept, and a few industrial actors were interested about this potential. So we started to make a first business plan. The plan was to sell pens at the beginning. And we started to participate to startup competitions, and this was really important for the beginning, because we didn't know how to start, and the different contests permit us to structure the business plan, to get the first amount of cash, and we started like this. So two important things, very important things, were the competition of Genopol, because we won a first prize, not the first prize, but the first prize of PILI. We won it, so I decided to work full time in January 2015. And then we got a lot of support from BPI France, because this activity can be key for the French agriculture, because if we succeed to convert sugar into dyes, we can then potentially transform other agro materials. Yeah. So it's very strategic for France, because we have a lot of agricultural production, so we can valorize this biomass into high value compounds. That's why we got a great support from the
That's people. Great. So just for those who don't know us, who don't know Genopol, so it's the biggest biotech incubator, accelerator cluster in Paris, and BPI France is a French bank supporting a lot of innovation, a lot of startups. From what you said, there's two questions I come out of it. One question is linked to, it's really a non-conventional approach to do it. I mean, especially without any tech, or without like developed tech, going out, pitching partners, pitching clusters, and trying to get a price. It's not conventional. So how did you manage to convince them without a tech or proper, like, I don't know, IP? How do you manage to convince them?
Yeah, it was quite difficult, because of course we started from scratch with no IP and no any technology. So on one hand, it's difficult because you have nothing. So you have a little bit to
at least you have nothing to lose.
Yes, yes, sure. So you at the beginning you have to not to fake it really, but yes, you have to be really pushy on what you could do if people help you. But on the other hand, now we are developing a technology, and we are 100% independent. And this is a wonderful asset, because we are not dependent of for now of any university or any corporation. So at the beginning it's very difficult, but now it's an asset. And how did we succeed to convince people? Well, it was really difficult, but one thing was very important. And from the beginning, we developed this technology because people wanted. People were interested in this technology because the potential is clear, is to produce colors not from petroleum but from sugar.
Yeah.
And the potential is here to make it in an efficient way, so we can be competitive, and the process is very cleaner. So,
actually, that was the second part of my question. Actually, I mean, the second question was, which I think is great, is that you, I mean, actually, you try really to solve a problem instead of having a tech and trying to find a problem to solve. You started the other way around, which is more business way to do it. You look at the market and then you try to fix it. But it's a good example that you can also do that in biotech, and you can be successful in doing it. And now my question is, can you expand a bit on how big is that problem? How big is the problem of synthetic dyes? Yeah, how big could how big is the problem?
Yeah, sure. So the first colorant called Movein were discovered in 1852. And before that,
for plants.
No, in the Movein in 1852 was produced by petrochemistry, and before, yeah, all the colorants were produced by plants. For instance, in France, in south of France, you have a plant called Garance, which produces red pigments that were used to dye the street of the Napoleon army, for instance. And the problem with plants is that the color range is limited. You have to use often you have to use toxic chemicals, mordants, to fix the colors. And above all, if we want to produce colorants from plants, now it's impossible, because it needs a huge surface to grow all these crops. So in 1852, a researcher find this first Movein. It was an accident, but then he discovered many ways to produce colors, and since then 99% of textile colors come from petrochemistry, and it's the same for plastic or ink, for instance. The problem is it's fossil based, so it's petrochemistry. So the raw material is a fossil material. It's polluting to extract, and the process to convert this fossil source of carbon into colors is also problematic and very consuming in terms of energy. And especially in Asia, where those colorants are produced and used, there is a big issue, because now it's changing, because the Chinese government become very strict on the waste treatment. But for a long time, there was a very huge issues about pollution of industrial water, and very big issues for environment.
And I guess in India it's the same problem.
Yeah.
Or Bangladesh.
It's a little bit changing now, but you have a problem of sometimes of toxicity of these colorants, but not all the time. Sometimes it's a byproduct of petrochemistry that are toxic. That means that you have the colorant, but you have some kind of impurity coming with the colorant. And because of the chemical steps, you can find also toxic impurities within the dyes.
All right, and just to also to understand better also how you will solve that problem. And from my understanding, you are trying to produce exactly the same dyes as chemical synthesis, the same properties, so you can replace it inside the actual process. So at the end, if you ship it to factories in India or Bangladesh and they don't recycle the color, it will end up in the same rivers.
Yeah, sure, but yeah, this is something. The first thing is that we are developing only safe colors. So on the colors we are developing, we make ecotoxicological tests in order that it's not polluting for the environment. So this is the first point, very important, that we are not developing toxic molecules. In the past, chemists didn't make this kind of tests all the time because it's costly. So now it's systematic. You have to develop this test for every new product you develop. So this is the first point. And the second point is, yes, of course, dyeing houses have to treat their water, but not only for colorants, because at the end, colorants are not especially the more polluting molecules in the waste water, because you also have you can have a lot of salt, which is very polluting when there is a high concentration, and a lot of different chemical auxiliaries. So the treatment is also key.
Okay. And I guess the color is the most visible one, so it looks impressive on pictures, and you have the whole river that is blue. I mean, it's but this is the most polluting. Okay, I see. Yeah. And as you really try to replace the production of these dyes, so from instead of chemical production, from using oil or petrol to using sugar with microbial fermentation to produce exactly the same color. Did you do any life cycle analysis to compare the two production methods? And if so, how more effective is your method? Because producing biologically sounds like it's always better than chemically, but I mean, you still need a lot of water, you still need a lot of sugar, you still need probably some chemicals as well to purify. So how's it different? Can you guide me through that?
Yeah, sure. No, it's a very important question because producing biologically, it's not in all cases more efficient, and also you can produce biomolecules that are toxic. So bioproduction is not 100% win solution. We are currently making a precise life cycle analysis with a third organization. Yeah. But what we can already say is that the difference between the chemical synthesis and biosynthesis is very high, because the key figures is for 1 kilogram of colorant dyes, you need 100 kilogram of petroleum, you need 10 kilogram of chemicals approximately, and you need between 1,000 and 8,000 liters of water.
Okay, and how much do you need?
We need only 200 liters. So we reduced by 5 the the amount of water you need, and we don't need the 100 kilogram of petroleum and the 10 kilogram of chemicals. So we'll need between 10 or 20 kilogram of sugar, approximately, to produce this kilogram of colorants.
So basically, a factor of between 10 and 50 times less polluting, basically, for the production.
Well, it's very complicated question, because you cannot compare all the products with the same indicators. So you try to make some correlation, or to use, for instance, the CO2 as the main CO2 emission as a main indicator, but you cannot compare CO2 emission with, for example, toxicity. It's just impossible.
But basically, but from the numbers you said, it's basically 5 to 10. I mean, at least on the water, which is one of the main resources as well. Okay.
So what is very important is approximately 4 indicators to reduce the water use, to reduce CO2 emission, to reduce the amount of energy you need to produce the same amount of colorants, and 4, to reduce or avoid toxic byproducts. Because often it's not the colorant itself, because a lot of toxic colorant has been removed from the market, at least in Europe, but it can be the byproduct from the synthesis that is toxic.
I see.
So that's why, because biotech is a very powerful solution.
Makes perfect sense. It's makes and now we can go a bit deeper in the technology, and then how you precisely produce that color. So I guess you're using some forms of your own strains, some form of enzymatic engineering, I guess some forms of gene editing. Can you describe a bit more how you make it work? What's the magic?
Yes. I'm not biologist, but I can explain simply. So yes, there is a metabolic engineering in order to optimize the pathway between sugar that is decomposed and recomposed within the cell in a molecule which is a colorant. So we work on this path and make it more efficient, and the game is here. So we have a dedicated team working in Toulouse on this topic. So there are now 6 persons in Toulouse, and we work with all the classical genetic tool to make this cellular factory very efficient.
So CRISPR, let's say CRISPR, then high throughput screening to optimize the process, and all these kind of things.
Well, what I know is that CRISPR or high throughput technology are not the best way to work. It depends. From different cases, but for us, we don't especially need these tools for all the modification we make. High throughput can be very interesting in let's say in an optimization phase, but it's not. I think it's not the great way to work during all the research and development phase.
Okay, makes sense. And so you're using bacterial strain.
We work on classical industrial strain.
So not bacterial, yeast. It's not public.
It's it's all that I can say.
If you cannot say this, okay. And so, can you say where do you develop the IP? Is it like on the metabolic pathway you developed? Is it on your strain? Is it on what you put the IP?
Well, it's important for us to protect the IP, especially regarding the products. So first, the IP come from products. It means the molecules and their applications. Of course, biology is also important, but the most important for us is located in molecules and application. Because at the end, we can be defined as a biotech company, but which is very important is that in fact we are a biochemistry company, because our goal is to provide biomolecules. So the chemical approach is very important, and this approach is completely leading our strategy and our development. Biology is a mean to produce in a more sustainable way and more efficient way, but at the end we work to develop biochemical products.
Yeah, your product is the dye. It's not the process.
Yeah, exactly.
Yeah, I see. That makes sense. And so, yes, I guess one of your upcoming challenge right now is also the scalability and how you will produce big batches to then optimize the process, and then having a cost-efficient process, and then being able to sell these dyes at a competitive price as well compared to the chemical equivalent. So how advanced are you in the scalability? Yeah. How challenging is it?
Yeah, sure. For now, this is not our main activity. Our main challenge is not the scalability. For now, the challenge is to develop a pertinent product portfolio. So we have to select the best product and select the best market. And this take a long time, because we have a lot of validation tests to do in different conditions. It's not enough to just produce a biodyes from fermentation. Then you have to work on this product, to formulate this product and adapt it to several applications. So it's very different to put a dye on a textile, or and it depends also about the fibers. So dyes for let's say for cotton are not the same as dyes for polyester, polyamide, or for wool. It's completely different. So we are working on the product development right now. The scale up will come later, and we think that it will not be a big technical issue, because there are already a lot of process that have been scaled. There are some challenges, especially on the downstream process, because it's a large part of the production cost. But right now we are focused on product development.
Yeah, I mean, I guess until you find the right product, you cannot move to scale up. Okay, makes sense. And then one of my last question is more on the stakeholder in the dye industry, or also your competitors. How do other dye producer look at you? And on the other hand, how does other bigger industrial biotech actors, I don't know, like Novozymes, these kind of guys, look at you? Like, what are your discussion with them? How do you place yourself inside this whole stakeholder ecosystem? Let's say it like this. Let's go like this.
Sure. Well, there are a lot of big players are interested about PILI technology. So we have very interesting discussions with them, chemists, but also big biotech companies. And yes, the discussions are very interesting because we are not going to fight with them. It's just impossible. We are in a
You're not trying to disrupt them.
No, I think that we will disrupt the market with them, because the goal is to put our products on the market and to make to change the way we produce these colors. So we are very open to discussion. We are discussing about them with them about potential partnerships on distribution, but also on production. So our goal is to make the best the best deal to put our products on the market as fast as possible.
So you didn't sign a partnership yet?
No, not yet.
Okay.
But discussion and test are starting now.
And how much time do you have until signing that partnership from the fundraising? Or people running out of money?
I would love to have the answer. So what we are talking about making first collaboration to see what is possible and what's the potential, and I think we will be able to make an official announce maybe next year or in 2 years. I don't know exactly.
Depends on the partners, I guess. Yeah, sounds good. Looking forward to that announce. My last question, a bit more open question on the do-it-yourself bio community. I think it's an extremely interesting community, and when you look at it, it's born basically in 2003, 2004. Early 2000s, and the development it had over like less than 15 years is pretty amazing. Now you have biohacking spaces everywhere on the world, and I think you're a good example of how bringing people from different disciplines into one space can create a lot of, let's say, call it value or solving problems, or a lot of interest. So yes. What's your view on the field? Like, what do you see coming in the next 5-10 years in the do-it-yourself bio field? Do you believe in the parallel between personal computing and personal biotech? Can you tell me your view of the field?
Yeah, sure. I don't really see a correlation between personal computing and individual biotech or doing bio, because biotech is very, very hard to develop. You need a lot of equipment and expensive equipment. If you make like PILI, if you want to make real products and produce and bioproduce something, so it's very different. But what I see, what in terms of movement, which is very and potential, which is very interesting now, is the gathering of different competencies, and now it's very much easier than before, because people can easily for instance, when we started PILI, I was studying in Barcelona, Spain, and we were always working on Skype with our other co-founders. I don't know if 10 years before it was possible. So what I see is the connection between different people, which is very more easy today, and the fact that people are also very mobilized to help if we develop clean solutions. So now you have to realize that PILI succeed to raise money not because of only its co-founders, but because we have been helped by I don't know, maybe 50% that has a key role in one step of our development, and it's incredible. So what I see is a connection between people, which is now very efficient, and also the system, especially in France, which is very which is very helpful to launch this kind of project, because two things: because you can have support from BPI France or other public organization, and it's a huge help, because if you don't, if we didn't win so much prize,
would be hard.
Impossible. Yeah, very hard. BPI France is with our historical shareholder SOS Venture. BPI France is the first investor, in fact, in PILI from the beginning. And also in France, you have an ecosystem of technical platform, which is exceptional. So where we are based in Toulouse, you can find a few places like this in Europe. It's very exceptional, because you have access to a lot of expensive materials, and also excellent expertise in the field of molecular biology, fermentation, downstream process, etc. And this is a key enabler, because if we didn't have this platform, we never succeed.
No, that's great. Yeah, Toulouse White Biotechnology is an accelerator / incubator in Toulouse, which is really great. I recommended it like last week to a SynBio founder in Berlin, actually. So it's funny. That's great. Thank you, Jeremie. We are running out of time. Thank you very much. And just to mention that you were on your phone and it worked. I don't know which is your carrier, but thank you for your nice mobile network. And good luck for the coming months, for investing and burning all that money, and make a nice party, and see you later. Thank you.
Thank you, Philippe. Bye.
Bye.